Hi, my name is Brianna Oliphant, and I am in Group 4 for the protein modeling assignment, and my assigned protein is the melanocortin 1 receptor, or the MC1R protein. This is my 3D model of my protein. This is the starting point of the protein, and this is the ending point of the protein. The blue is the cell membrane, and you can see that the protein is a transmembrane protein, so it kind of goes like in and out of the cell membrane, with this area up here being the outside of the cell and this area down here being the inside of the cell. This is the starting point of the protein. You can see this is the ending point. As I said, my assigned protein is the melanocortin 1 receptor, or the MC1R, and the mutation is when arginine number 67 is replaced with cytosine, and the mutation doesn't change the overall shape of the protein. So I just marked here kind of where the mutation would be, this dot right here, and this is where in the chain the different amino acid would be. So the normal function of the protein, this protein is a receptor protein, and it binds to an melanocyte stimulating hormone, and the hormone is called alpha MSH, and the hormone causes pigmentation to be produced in, well, for these purposes it's beech mice fur, but it's usually in mammals, and it causes pigmentation in the fur and skin. Like I said before, the MC1R is a transmembrane protein, so it's usually found in the cell membrane. The cells that it's found on are called melanocytes, and as I said before, this protein is found in mammals. The mutation makes the protein function differently. So like I said before, it doesn't change the shape of the protein, but it does have kind of an effect on its function. When arginine 67 is present, when the protein is not mutated, the hormone binds to the protein, and the protein sends a signal through the cell to produce eumelanin. Eumelanin is like a darker pigmentation, but when arginine is switched out with cytosine, less eumelanin is produced, and something called pheomelanin is produced, which is a lighter pigmentation. The reason why pheomelanin is produced is because cytosine isn't as stimulated by the hormone alpha MSH that binds to this protein, which causes the pheomelanin and lighter pigmentation. And that lighter pigmentation is the reason why this mutation in beech mice causes lighter fur colors instead of darker fur colors. So the main functional groups present in the original amino acid side chain: arginine contains an amino group, a carboxylic acid group, and a side chain consisting of 3 carbon aliphatic straight chain ending in a guanidino group. Arginine side chain is polar, which means that it's also hydrophilic, and it is a positively charged side chain. So it is a positively charged side chain. Compared to other amino acids, arginine is a little bit on the longer side, and arginine can also be considered amphipathic because part of its backbone is hydrophobic, meaning that it could have reactions with both hydrophobic molecules or amino acids and hydrophilic molecules or amino acids. Arginine can make hydrogen bonds, and as I said before, since it's amphipathic, it can interact with both polar and non-polar substances to some extent, and it is also a positively charged side group. Again, that is going on right here where this little purple dot is. So the new mutated amino acid side chain I just talked about was arginine. So now for cysteine. Cysteine is a triprotic acid with 3 ionizable functional groups. It has a carboxylic acid, an amino acid, carboxylic acid functional group, an amino functional group, and a sulfhydryl functional group. And cysteine also has a side chain that is polar, but it's not charged. Compared to other amino acids, cysteine is a little bit on the shorter side, but it's not the shortest. It's still bigger than a few other amino acids. The difference in the types of bonds that they can participate in. So cysteine can form hydrogen bonds like arginine can. However, cysteine can form disulfide bridges when there's cysteine dimers, and the disulfide bridges will link them together, and then cysteine can also form covalent bonds with other molecules. So as stated before, the mutation in this protein doesn't affect the actual shape, and it also doesn't really, the protein will still function with the mutation. However, the outcome is of the signal sent through the signal transduction pathway of this protein is different. Like I said before, due to the fact that cysteine isn't stimulated by the hormone alpha MSH, the mutation causes pheomelanin to be made instead of eumelanin, and a lighter hair or even lighter skin color as a result, instead of that darker pigmentation that you'd get with eumelanin. There are limitations of my model. Obviously, it's not as detailed as it could be because I don't have the equipment to imitate microscopic things such as binding sites and the entire signal transduction pathway. This is just the protein, but the entire process goes on throughout the whole cell, and there's second messengers and also other things along that pathway that trickle down to produce the appropriate response to when the hormone binds to this signal molecule. So obviously my model doesn't show all of that. It just shows the protein, which doesn't change shape. So really my model doesn't show the full extent of the mutation, if that makes sense. It just is this one amino acid that changes. And yeah, just my model's not as detailed. It doesn't have the site of the actual binding site between the hormone and the protein, because like I said, I can't imitate that because my protein is overall just like too big. Like the whole thing is too microscopic to actually imitate. And yeah, that's my presentation on the MC1R protein and its mutation when arginine is replaced with cysteine. I hope you liked it. Thank you.